We have one more example. And then after this example, I can go through more of the PK data for the other people that have asked to see examples for those. So for this example, instead of going under the adverse effects, we'll go under drugs. And basically, the question is, a customer was interested in finding out what adverse effects have been observed in monkeys specifically who were administered disease-modifying anti-rheumatic drugs. And the follow-up question to that was, did any of those drugs cause abortions when given intravenously? So if we go under drugs, we can select for the disease-modifying anti-rheumatic drugs right over here. And again, what's nice here is that we get a list of all of the drugs. We can see also from this view pharmacokinetic data, if we're interested in seeing pharmacokinetic data from here. We also get a list of all the targets which are related to those drugs listed. And then just below that, we see all of the adverse effect toxicity data. So the question related to abortions. So all we have to do is scroll down, and we see pregnancy. Click on this, and we get abortions and stillbirths. So for this, we can go under the preclinical tab because we're interested in looking at was this present in monkeys. And now from here, we can use the filter, which is really convenient. So we go under species, we go under vertebrates, mammals, primate, and we get data for monkey. We click filter. And here you can see how abortions were observed when giving the intravenous route of the drug. So it's possibly something to be wary of if you're conducting studies with this drug class, that this is a side effect that you need to be careful of. So those are some ways in which we can actually find safety data using PharmaPendium. Very easy to use and very user-friendly. Now we will go to the rest of the 25% of the audience who requested to see more PK examples. So let's just go back to the home page. So let's start off with a more simple example. One customer had asked us, show me all the drugs in my class of anticonvulsants with a half-life of 5 hours or more. So in that case, all we have to do is go under the pharmacokinetic tab. And here it says we can add drugs or a drug class. So we're going to add a drug class. We also have the option of adding a specific target. So that's also available to you if you have the product. And we're going to find anticonvulsants. And we're just going to click Add. And what's nice here is that you can actually narrow down your search results if you're interested in doing that. You get a list of all of the drugs available over here. You can click Remove if you're not interested in finding data related to that particular drug. And then here we have the PK parameters that are present in PharmaPendium. So for example, we have absorption, which shows all these parameters. We have binding, which shows all of these parameters. In particular, protein binding is quite popular because it's data that's very difficult to get. We have biotransformation data, data related to distribution, and data related to elimination. So the question related to half-life and finding drugs which have a half-life of 5 hours or more. So all we have to do is click on the half-life tab, select it, click above, and then type 5. We go back to the top, and we click Search Now. And then here are the results that we get. And what's nice here is that it really shows you the drug name, the species, the study group, the dose that was administered, the route of administration, the parameter value, standard deviation, time, and then concomitant. So you can actually see differences between, for example, fed and fasted states. Now, as an example, let's go to the top and click on, for example, this link that says bioequivalence review. When we click here and we actually scroll down a little bit, you can see that we get actually some really interesting data which can help you set sort of initial standards for any experiments that you're going to conduct. So this example is a very nice, simple example that shows you what kind of data you can get access to if you do have PharmaPendium. So let's go back to the home page and do another example. So one customer had asked us, show me all tissue-specific PK data for drugs interacting with epinephrine targets. So in order to answer this question, we could actually go under the Targets tab. Here we can type in epinephrine. We click Go, and we see a tab for epinephrine targets. We select this. Again, what's nice here, you get a list of all of the drugs. And then over here, you have the tab, as we previously showed in another example, which says View Pharmacokinetics Data. So you can click here. And now under the filters, you can actually select for tissue specificity. You can say I want only tissue-specific results. And you click Apply Filter. And you'll see that the tissues are actually described in the parameter. So it'll tell you the parameter and then the tissues that are relevant. And if you scroll down, you can see there's quite a lot of breadth of data which is provided to you. Again, this would take a very long time to find out if you did not have PharmaPendium. And then as one more example, we can, for example, look at the drug. What if we were just interested in looking at the drug aromacin, a breast cancer drug? What would that result look like? So we type in aromacin, just waiting for it to load. And we get all results which state aromacin in it. Here also we get a direct link to the drug page. And here we can go directly to, for example, pharmacokinetic data. Now let's say we want to look at the clinical data, so only data that's available in humans. And let's say we want to look at protein binding. So here we go under Show Filters. Under Parameters, we see all of the PK parameters that we were interested in. So let's go to Binding, Protein Binding. Let's say plasma protein binding. We can click Apply Filters. And here what's nice is that you can actually see for aromacin, if you look at the route of administration, there's two routes of administration, oral and in vitro. And for both of these routes of administration, we see that the parameter value is greater than 90%. Knowing this information, you might want to know what is the value in preclinical studies, and what is the right animal model that I need to use in order to simulate this result in humans. Well, to do that, you can go under preclinical data. Again, we can filter our results for plasma protein binding. And here what's really nice is that we really get a black and white view of what preclinical model is relevant for us in studying plasma protein binding studies. So for example, if we look at dogs, we see that there are two routes of administration, in vitro and oral. And we see that for the in vitro studies, this parameter value seems to translate well in humans, but the oral studies don't. And in fact, if we scroll down a bit more, we can see there were studies done in monkeys, mice, and rabbits, and all of them used in vitro routes of administration, probably because it had the best way of simulating the same conditions in humans. Now, there were additional studies done in rats, for example, where they did use oral and in vitro studies. And here we can see, similar to the dog, that the oral studies don't translate, but the in vitro ones do. So this can be really critical information to know when you're designing your preclinical studies, is what is the right animal model for me? And using PharmaPendium as a tool, you can actually get that answer extremely quickly and then plan your studies accordingly. So those are about 6 examples which we have presented, 3 related to safety and 3 related to pharmacokinetic data, which you can then use to sort of advance your studies even further and even plan them at the very beginning before anything starts to go wrong. So with that said, Amo, I'll leave it to you and we can start the Q&A session.
Fantastic demo, Pooja. Wonderful. I love this last one on setting up your preclinical studies. Really clear examples. Thank you. And we do have a couple of questions that have come in. We have one going back to, I think, your safety example at the beginning, Pooja. And they're looking for more information on showing all adverse effects and toxicity data that interacts with the same target.
Okay, perfect. So what I'll do is I'll go back to the home page. And if you're looking for all of the adverse effect and toxicity data for a particular target, again, you can go under targets. And you can type, for example, let's say we do 5-HT2. And we select 5-HT2 receptors as an example. We can get here, we get a list of all the drugs which target 5-HT2 receptors. And as we scroll to the bottom, you can see all of the safety and tox data in the preclinical, clinical, and post-marketing studies. So as an example, let's say we go under clinical. And we click on the first result. You can actually see a lot of the information that you're looking for. And here is where they describe the adverse events. So again, it's very easy to find the information that you're looking for right in the FDA approval packages. And again, this information is presented in a very unbiased view. You directly go to the original document. So that's a really nice way of showing how you can get to information which relates to the specific target that you're looking at.
We had a question on how to find phase 4 commitments for drugs in a particular drug class.
Oh, okay, perfect. So here actually you can get kind of creative using PharmaPendium. So for example, we can actually search with quotes. Now what we'll do first is type phase 4 commitments. And here we put it in quotes because we're looking for this exact phrase. And we can search this exact phrase in PharmaPendium. Now, if we did not have the quotes, then it would search the words sort of independent of each other. So let's see what the search results bring us. Okay, so here we have over 1,356 results. And you can see sort of the drug names listed here, where the source is. And what's nice, too, is that when you see the year here, you can actually click on the year. And you can get the most recent results first. So this is a nice way of getting the most recent data. Now, if we go under Show Filters, you can see that you can actually select by drug or drug classes. And it says over here there's a number listed beside each drug class that tells you how many hits are there per drug class. So that's a very nice way of doing that. Here we see we have, for example, an analgesic, 70 results. We can go here. and sort of get all the data that we're looking for all the different drugs that we see. So that's a really nice example of how you can actually even get creative with PharmaPendium and type in things like phase 4 commitments and see what you get. We've had other customers which have typed in, for example, reviewers' comments and then filter according to a specific drug class to see what went wrong in drugs that were approved in the past and how did the people then plan accordingly to address the FDA concerns. You can also see with reviewers' comments what are sort of the new emerging concerns that FDA is having for a particular target or for a particular drug class. So that way you can stay really recent and be as best prepared as possible when, for example, if you're in the preclinical phase and you're getting an IND approved, you can then do your best to try to be as prepared as possible to get the best clinical trial designs so that you minimize any surprises that you may get from the FDA.
Super. And what we're seeing, Pooja, of course, through your examples, is the many ways that you can start your search, the many entry points in PharmaPendium, depending on the information that you're looking for. And we have time for one other question related to that. And we have someone in the audience asking about searching by chemical structure.
Perfect. So here, again, I'll just go to the home page just to refresh that search. And here you have chemistry search tab here. So there's an advanced tool, there's a chemistry search tool, safety data search, and pharmacokinetics. So we'll go under the chemistry search tool. And here, again, you can click this structure box to create or edit a structure. And then you can search accordingly to see what drugs pop up. So this is one way in which you can use this chemistry search tool to then do more advanced searching. If anybody requests a specific example regarding the chemistry search, we can have our senior product manager send over a file to you so that you can get a better idea of how to use this chemistry search feature, which has been quite popular with customers in the past.
Okay, great. So, if you would like more information or a specific example, then by all means contact us after the webinar with the email address that you'll see in the follow-up emails. We'd be more than happy to hear from you. So, only left for me to thank our audience, most importantly for attending. We hope that you have seen many interesting examples today that will keen your interest in PharmaPendium. Thank you, Pooja, for your excellent presentation and demo. Today's session offered an introduction to PharmaPendium, and we're also offering more in-depth sessions throughout the year. The next online PharmaPendium session will be on the 29th of May, and this session will offer a more in-depth view on pharmacokinetics, which Pooja did touch on today. If you'd like to learn more about that, please do register for our next webinar. You will receive the details for this in a follow-up email. And of course, feel free to register for as many PharmaPendium webinars as possible, or indeed send it through to your colleagues. We'd love to meet your colleagues in the webinar as well. When you leave the session, a survey will pop up. Please fill out what your thoughts are regarding this webinar so we can aim to improve the webinars going forward and give you the information that you really want to see. Besides this, you'll receive the Q&A by email shortly, and we will replace a recording of this session on our training desk, and we'll make sure that you receive a link to that recording so you can play back in your own time. Many thanks.